Bismillahir Rahmanir Rahim. Ladies and gentlemen, I'd like to welcome you all for our live webinar on the iron deficiency and the immune system. In fact, this is one of the series been initiated by the Emirate Medical Association OB/GYN Society in order to gain a CME and new knowledge in different fields. I'd like to thank the organizers, and I would like to thank Saja for giving us the opportunity to meet and to see you all on this webinar, as well as on YouTube, which is really going live at the moment with our presentation. This is a webinar that is really streamed on MCI Middle East YouTube channel. The webinar will be available on demand on the MCI Middle East YouTube channel, and there will be an up to 1.5 CME accredited by the MOHAP. There is a live chat support, which is available on the WhatsApp during the event, and you can see the number there to record. Also, we're gonna have a question and answer session at the end of the two presentations for tonight. I hope we're getting there, and can see there is lots of candidates are joining us, and I think we are ready to proceed. Dr. Guy.
Bismillah ar-Rahman ar-Rahim. I would like to extend my thanks to the organizing committee for giving me this opportunity to be part of this interesting webinar. I think we should start now. The number of participants is rising rapidly as we speak. So in order not to be short in time, I always like these webinars to be like really like with a fixed time and really to the point, so that everyone can manage their time accordingly. Well, let me start by expressing the honor I'm having to introduce Professor Dr. Hisham Arab. Well, actually, I can go on for hours just enumerating the credentials of Dr. Hisham Arab. He's one of the esteemed and the figures and the eminent figures in the field of OB/GYN. He's a consultant OB/GYN. He's one of the figures in perinatology and minimal invasive gynecology in the Gulf area. He's director of Fetal and Women Health Program in Saudi Arabia, and he will give us his insight about the immunological component of iron deficiency, which is quite an interesting topic even for myself. Professor Hisham, تفضل حضرتك.
بسم الله الرحمن الرحيم. الحمد لله والصلاة والسلام على أشرف خلق الله محمد بن عبد الله صلوات الله وسلامه عليه. Good evening, ladies and gentlemen. I'd like to thank first EMA OB/GYN for their kind invitation to present to you this talk. I'd like to thank Saja and the organizing webinar company for this interesting and very challenging topic. Actually, iron deficiency and immune system, it's a new thing. The science of it is still developing, and I know we've been talking lots of about anemia, iron deficiency anemia, but to talk about the iron and immune system, I think this is need to be a little bit explored further, and we're still waiting for further studies on this area. Now, to start with, I'd like to start with this phrase, Bismillah ar-Rahman ar-Rahim, وَأَنزَلْنَا الْحَدِيدَ فِيهِ بَأْسٌ شَدِيدٌ وَمَنَافِعُ لِلنَّاسِ صدق الله العظيم, Ayah 25, Surah Al-Hadid. With this phrase that we have seen that God created a heavy metal on earth, and from our science, we have seen that iron is one of the most abundant element on earth and essential to almost all organisms. So when it comes to utilization or purpose of iron, in my view, and this is my own view in fact, I can see there are two arms for iron purpose or iron utilization. One is the hemopoietic, which we know, and that will lead to the anemia with deficiency, and the other one is the immunologic. And if there is a problem with that, then usually you're going to end up with some kind of immunological disorders. So iron is behind anemia and also behind immunological disorders. Variety of disorders varies from infection to tumors to various diseases. Of course, at the end, we're looking for health. We want to enjoy our health, and this particular aspect need to be really probed carefully. So this was the iron purpose. What about the iron source? Well, the only natural source of iron is diet, of course. We're not talking about therapeutic sources of iron, all right? So natural source is definitely diet, and here is very important. The iron comes inside the diet. The iron, this diet we consume need to pass through the stomach in order to extract the iron out of it, and also from there this iron can be passed into the intestine and then will be absorbed from the intestine. So here is a very important area that we have to take care of, that because we only take iron from our diet, we need to have a functioning stomach. Those who had surgery, the bypass or the sleeve, usually they have a problem with that, and that's why they don't have much iron in their system and not much iron being absorbed, and that's why they need to be supplemented carefully. So this is the source of iron. What about iron absorption? In this diagram, I'd like to take you in details a little bit here from on the introsite, because as the iron is extracted from the diet, from in the stomach, it passed into the duodenum and the jejunum. The introsite of the duodenum actually has this villous surface for each cell into the lumen. And here the iron can be presented as a ferric with a Fe+3 iron, or as a heme ferrous, which is Fe+2 iron. Now, it has to be absorbed here, which is we call the heme transporter across the villus or the membrane of the cell into the cell. This can be an apical transporter or a DMTA1, or the divalent metal transporter that can take just the iron, the ferrous iron inside. For ferric ion, which is the +3, need to undergo some kind of what we call a reductase function enzyme, cytochrome B reductase enzyme, to reduce it from Fa3 to Fe+2. So here now the ferrous is really ingested inside the stomach, and as a result, then it either get accumulated and either be accumulated in the cell, and it has to be stored in what we call the ferric cage or storage area, and here actually is called ferritin. So it has to be, again, it had to be converted into the ferric, which is Fe+3, and then accumulated within the ferritin molecule inside the cell. Or it can be transported into the plasma and into the circulation, which is the function in order to reach the bone marrow. And here there's what we call a gate, what we call the ferroportin. Again, the ferrous inside the cell needs to be converted to ferric, which is here we use the hepcidine. Now the hepcidine oxidize the Fe+2 to Fe+3, and then the ferric comes out and picked up by what we call the transferrin. So the transferrin is the molecule that carries 2 ferric ions on it and goes into circulation all the way to the bone marrow. So this is how the iron is being absorbed from the enterocytes. Then comes to the arm 1 I was talking about, the hemopoietic purpose, because 75% of absorbed iron is used for erythropoiesis. All right, that's a large percentage of it. So what happens, as I mentioned before in my previous diagram, that the transferrin is carrying now the ferric into the circulation. Where does it go? It goes to the bone marrow. Here, of course, what we have, the erythroid progenitors. The erythroid progenitors, they have receptors on them for the transferrin. All right. So as the transferrin sits on these receptors, then these iron get inside these erythropoid progenitors and deliver the iron to the erythrocytes. Very good. So here hemopoiesis started and created the red blood cells rich in iron. But at the same time, those RBCs get old. So what they happen, really, they die. What will take care of that? The macrophages. Here is the macrophages, is the one that engulf those RBCs. Those RBCs, they have iron. So again, inside the macrophages, there is the mechanism of extracting that iron from these RBCs and stored into the ferritin, or again exported across the ferroportin gate I mentioned before, like the one here. Again, it's the same thing here, into the circulation, into the transferrin. So the same here, the macrophages working is the same purpose here as the enterocyte. The enterocytes gets the iron from the diet, from the lumen, while the macrophages get the iron from the dying RBCs that were ingested in those macrophages. So again, the cycle continue and the bone marrow benefits. So that's why I'm saying it's a hemopoietic purpose of this iron. So just to simplify this in writing or just in few words, the iron metabolism in general in healthy individuals actually is largely found in intracellular, and the iron is intracellular in any cell, can be found in the form of ferritin. So it's not just in the enterocytes. Any cell in the body has ferritin and also has what we call the cytochromes and FES proteins across the body. However, in the erythrocytes, the iron exists inside the heme of the hemoglobin. What about in the circulation? In the circulation, the iron exists into the transferrin. So these are the areas, whether intracellular or circulating iron, where it exists in our body. It's very important to note that there is a compound called the ceruloplasmin, which is really rich in copper. 95% of copper exists in this enzyme, and the purpose of this enzyme actually exhibit a copper-dependent oxidase activity, which is really work as oxidizing the ferrous into the ferric form in order to make sure that the transferrin or the lactoferrin in our system take the iron and carry it around in our system. Just a small word about the ferroportin, which is really expressed in most of the cells again of the body, because it's the one that really pushes that ferrous outside the cell into the circulation, and which is really contributing to the plasma ion levels, and that's why it can be found in the intestinal enterocytes, in the macrophages, and also in the hepatocytes. Another important concept here I want you to also to keep in mind, along with the iron metabolism, is the hepsidine. Hepsidine is also another hormone or enzyme produced by the liver, and its function is to block this ferroportin gates, so it reduces its expression, so they get internalized, and that's why there will be no iron released in the circulation, and as a result we're going to have a low plasma iron or iron deficiency anemia because of the lack of iron. So keep that in mind, where the hepsidine is working. Now, this actually the area that triggers the immunological purpose of the iron, because we have found that the increased hepsidine within hours of an infection of inflammatory insult, resulting in dramatic reduction in plasma iron levels. So somehow hepsidine is not just working on its own, but it is a reflex of an immunological reaction, of an infection, and why it will take away or stop anything iron that's coming out of those cells. They stop it from there. They keep the iron inside, so the circulation will be lacking iron. Why is that? Well, eventually restrict the availability of iron not only to the individual, to the invading pathogens, but also to developing erythrocytes in the bone marrow, leading to the anemia. So, and this anemia is called iron deficiency anemia, or anemia of inflammation, or anemia of chronic disease, because what we see here actually, yes, it's a protective effect, so that the iron will not be taken by the invading pathogen. Well, Yes, pathogens, invaders, they need the iron also, not just us. But unfortunately, as a result, we'll also suffer in cases of infection. So restricting the systemic iron during times of infection and inflammation represents an essential defense mechanism against pathogens, but under the pathophysiology conditions can lead to anemia and further severe pathologic resulting from the reduced RBC developments. So this is how the iron is being utilized in our cells. And I call it ionology. This is my own word, my invention, basic ionology. And here you can see that the iron, yes, be taken with RBCs, can be taken by the neurons and muscles, but also look at various immunological cells. The T cells, the macrophages, neutrophils, and the xenophils, all these, they're, making use of this iron in their immunological arm. This is the immunological purpose of the iron here. But don't forget, we have also pathogens, and the pathogens rely a lot on iron in order to produce their virulence in our body. So, in order to understand this, I thought maybe let's take a little bit from basic lessons in immunology. Here is what we call the humoral and the cellular immunity or cell-mediated immunity that we learned in our medical school. As you all know, the humoral immunity is relying on the beta cells, which is really producing the antibodies that will actually attack or result in some kind of neutralization of the organisms, or lysis, or even phagocytosis by the distracted organism by these phagocytes. However, for the cell-mediated immunity, we have here what we call the T cells, and here the T cells actually can develop some kind of two types, that the killers and the helpers, and they produce also what we call the cytokines and the chemokines in order to attack these pathogens, and then will be engulfed and taken away from the circulation. So simply antibodies and then cell-mediated and cellular immunity. So this is what happens actually when as a result of this recognition. That's what we call it, the adaptive immune system. Adaptive, that means this is what happening every day, every single second in our body. Whenever it senses anything, or odd thing, an organism or something odd coming into our system, these actually weapons, they just come into action and start to defend our system. And here the first thing happens, the T cells together with the B cells are the main components of the adaptive immune system. And here we can see that the T cells divided into cytotoxic and helper. The CD8 cells actually that are the cytotoxic. These are themselves actually kill the infected organism or the tumorous cells and release cytotoxic mediators such as the perforin. However, the T helper, the other CD4 type of T cells, is not really the one that kills, but it helps. That means it releases something which we call the cytokines or the chemokines into the circulation, and these are actually the one that stimulate other type of inflammatory cells or white cells in our circulation to do its job. For example, it affects the neutrophils, the macrophages, killer cells being stimulated also, and also the B cells, so it will be reducing what? Produce the antibodies. And of course, here what we see that there is an iron here required in order for these actions, in order for these cells to proliferate, in order for these cells to produce this kind of cytokines or change in shape or its activity. It also needs iron in its mitochondria, because this proliferation and this action need energy, and the energy comes from mitochondria in the cell, and the button that presses that energy production in the mitochondria is actually the iron. So, what happens in the macrophages then as a result of being stimulated from the T helper cells? Macrophages, another immune cell of the innate immune system, and present actually a foreign antigen to the adaptive immune system, the T and beta cells, to activate them. So they're helping also. They said, oh, we have something, an invader here, and they bring it to the T cells in order to be activated. Now, macrophages and Kupffer cells actually scavenge the heme from the damaged or aged RBCs in the bone marrow, liver, spleen, and extracted the ferrous, actually, ion using the HO1 system, and then either export it back into the circulation through the ferroportin stores or in its ferritin intracellularly. As I mentioned before, this is just how it is. The macrophage is really doing its job, and for the sake of utilizing the RBCs and release the iron again in the circulation. However, also the macrophages infected with intracellular bacteria induce programming of the iron metabolism to limit the intracellular iron and availability to these. So what happens? Usually there are two programs ready within any macrophage, and these relying on the ion itself and how it is really polarized, whether it is pro-inflammatory or anti-inflammatory. The M1 macrophage pro-inflammatory, what does it do? It displays an attenuated ion release and enhanced ion uptake and storage as an antimicrobial effect. So what it does actually, it does not release the iron, and so it keep it to itself, and it keep it in its ferritin component. So then the organism, whether inside the cell or even in the circulation, will not get iron. That's what this iron macrophage is doing. The M2 and what we call the anti-inflammatory pro-tissue healing, these actually, they work near the repair system. So what they do, they go not near the organism, but they go near the damaged area and release iron there through the ferroportin gate, again in the ferric form, and then this iron will be gulfed or taken by these tissues, undergoing the tissue repair. So really, it attacking at the same time, it's helping building. So here, this is how the macrophages, according to its polarization, do its function. Now, the hepsidine during the times of infection proven ion release in the circulation, prevent ion release in the circulation, which would otherwise fuel extracellular pathogen growth. Of course, the bacteria, when it comes into our system, it really, the first thing it does, it starts looking for iron. I'll show you how does it do that. And the hepsidine, that's why it stops iron from going into the circulation, so that those bacteria will starve and die. Now, the other cell that I need to discuss here also, which is very important in our immediate system, is the neutrophils. Now, neutrophils are the most numerous white blood cell in the body, and often it is the first responder at the scene of an invading pathogens or any tissue damage. Now, what does the neutrophils do? Number one, neutrophils secrete what we call the lipocalin-2. Lipocalin-2 is an enzyme to interfere with the bacteria scavenging host iron from the bloodstream. Okay, bacteria go in the bloodstream, it start to look for iron, for transferrin. They want to take it. Now, lipocalin-2 stop that mechanism to take place. Okay, so the primary function in host defense with when they encounter microbes is to ingest them into the phagosomes, undergo rapid burst oxygen consumption, and release antimicrobial proteins from the granules into the phagosomes to kill the microbes. So one is to lipocalin, so the bacteria will not get the iron. Number two, actually attacking the iron through its granules. You know, they have the multigranules cells. So this granules has actually what we call the ROS system, which is using again the iron to produce oxygen radicals. And these radicals actually will cause the damage into those bacteria, and really bacteria will die. And then we have also the superoxide radicals, which is just I mentioned, that help inside the neutrophils to kill those bacteria within them. Then we have also something called the myeloperoxidase MPO. This is an enzyme help in this process, and usually it has a green protein component, and that's why whenever you see a pus, okay, it's a bit greenish. This is the result of the multiple use or the extra use of this enzyme in that killing process. So anyway, whether it is a hydroxyl or tyrosyl radicals, these are actually orchestrated into the neutrophil to kill those organisms, and that's really attacked by them. Now, one important thing, also a function of the neutrophils, is actually producing a large amount of actually what we call the lactoferrin as well as the calpoprotectin. And lactoferrin is actually another compound that's been that holds the iron inside it, and usually scavenge iron from circulation, and it helps also to attack the infection. And I'll show you how does it do later on in one of my slides. Now, let's come to the organism. Let's come to any bacteria, how these bacteria go after the iron in the circulation or in the cells and try to gulf it, okay? To gulf these iron, they have like 3 or 4 mechanisms. Actually, it's just like a war, and I'll show you how this war really between us and the bacteria. First of all, these bacteria actually synthesize a high affinity ferric acid chelators. Okay, so chelators, that means is something, there is iron there, it chelate that iron, it takes it, stealing, all right, to the ferric iron, and it is ferric. I mean, reducing it to ferrous is another story. That's the bacteria tell you, I'll take care of that. But I know you have all the ferric going around in your circulation, so all I need just to steal that iron. So these are the chelators that take this iron from our circulation. Another thing is the lactoferrin or transferrin binding. So as you can see here in this diagram, this is the bacteria, for example, and this is the cilia here coming around it. But what does it do is actually it produces receptors that actually specific for any molecule that carry iron. So for example, we have here the transferrin receptor, here the lactoferrin receptors. It made also the hemophore receptors or the hematoprotein receptors. All these to gulp the transferrin, the lactoferrin, the heme, the hemoglobin, all these molecules that carry iron. So you see how much this bacteria is equipped and specifically attacking or going for the iron or any molecule that has iron. So that's why by the bacteria will having this system to take the iron, and then it has the system also to reduce that iron from ferric to ferrous, because the ferrous is the one that really does the job. So it has actually what we call the bacterial reductase enzyme inside it to reduce the ferric to ferrous. And I told you this is one of their function, to utilize that iron. Then is the story of the hydrophores. All right, here actually, as I mentioned before, we have the lactoferrin has 2 molecules, the ferritin and also that is transferrin. They have these molecules of iron inside it, or ions actually. So what does it do? They produce something what we call the hydrophores, which is really removing those iron from these lactoferrin and from the transferrin. However, our system actually created something what we call the siderocalin. Okay, the siderocalin is some kind of an enzyme in our defense system that comes against those siderophores. Okay, our system recognizes that the bacteria is sending siderophores now to take these transferrin and lactoferrin with the iron inside them. Now, what we did, our system said, no, don't do that. I have siderocalin. So the siderocalin comes to this siderophore to stop it from working. Now, what happened? The bacteria, as a result, start to synthesize something called the stealth siderophore. Now, here, the stealth siderophores is something to counteract the effect of this siderocalin. So it makes sure that its siderophore does its job. Now, it's not the siderophore, it's the stealth siderophore that actually ensures taking the transferrin, lactoferrin with the ion into its cell. So it is a war, and you can see here the bacteria doesn't really give up, and it starts to improve its genetic components and its ability to produce all these weapons, but at the same time we're also there for it. So when you talk about any cell metabolism regarding the iron, so in any cell, not just in the enterocytes, you can see here there is an iron uptake, number one. Whether it is ferric or ferrous, it has to go through the what we call the transporters, as I mentioned before, the DMTA1 or other transporters. Then the ferrous comes into circulation. So what happens here is inside any cell, there's a pool or labile iron pool, what we call it. So all in its ferrous, and this is how it works. Now the ferrous really goes ingested into the nucleus for DNA replication. This is very important. Or this ferrous goes into the mitochondria for the energy production. That's great. So this is how the iron is being utilized in each of our cells for energy production, for DNA replication. Is very essential. So what if we have an extra? Now, if we have an extra iron inside, unfortunately, there is a process here, what we call the radical oxygenation or expression of those radicals inside the cells, which can be very toxic. So this excess of ferrous is dangerous due to the ROS production, that can be really cause maybe damage to the cell itself. We don't want to kill our cells. So in order to prevent formation of these ROS, now what happens? The ferritin cage start to develop. There is a mechanism here where this ferrous transferred into ferric and kept inside this cage. But if we need it, then definitely this cage, through the action of the what we call the nuclear receptor coactivator number 4, the NCOA4, can really convert the ferric into the ferrous in order for the ferrous to be utilized, or to be, for example, transported across the ferroporting gate here into the circulation, and then the transferrin will take it. So this is how important for us to have the iron inside the cell. Now the iron accumulation can be detrimental to the cell, as I mentioned before here, and through the Fenton chemistry, the ferrous iron reacts with hydrogen peroxide to generate this ferric ion and also produce the hydroxyl free radicals. This is what we call the reactive oxygen species, the ROS, and these actually can damage DNA, can damage proteins, can damage lipids in our cells, and that's why we don't want these ROS. But remember, basal production of ROS is important for intracellular signaling, cell proliferation, and response to injury. So we need this ROS, the reactive oxygen species. We need them in a specific amount just to make sure the system is working and all these functions are carried on. But if there is too much of it, we'll have damage. If we have the appropriate amount, we have signaling for better purpose function. And here is just to give you an example of cardiac diseases. Those patients who have high incidence of ROS, the ROS production can contribute to the development of heart failure, cardiac iron accumulation, and comorbidities associated with like what we call the ataxia and hemochromatosis. Now, several chronic diseases such as cardiac, renal, neurological diseases, and cancer are also associated with the reduced iron in the circulation and a tissue iron overload, leading to the ROS production. So baseline iron chelating only prevent the conversion of less toxic ROS to more toxic ROS during the injury. Therefore, unlike antioxidants, if using strong antioxidants is not good because you will take all the ROS and then you leave your cells vulnerable. However, if you have a reasonable chelating agent, then it will just take the extra ROS and keep your cells functioning. I thought, let me talk about the lactoferrin, because I always mention about transferrin and lactoferrin. So lactoferrin, actually, as you all know, is produced by the mammary gland, actually, which is the human lactoferrin, which is a glycosylated protein consisting of 691 amino acids, folded into two globular two globular lobes. See, this is one lobe and this is one lobe. Now, the lactoferrin and transferrin have similar amino acid composition. Also, they have similar secondary and tertiary structuring of the protein so that's why they carry similar biological functions. The human lactoferrin and bovine lactoferrin are also similar to each other and have very similar antibacterial, antifungal, antiviral, antiparasitic, and anti-inflammatory and immunological activities. Now, lactoferrin binds one ferric ion atom on each of its two lobes. So you see this one ion, and there's another one in each one of its lobes. And how it does not release its ion even at pH 3.5. So this ion is really linked to the lactoferrin even in acidic media of pH 3.5. Why are we talking acidic? Because any infection area, usually it is acidic. So, I mean, it's still in an infected area, it will still hold to its iron to not be released to the bacteria. So, lactoferrin is therefore an important player in preventing bacteria from acquiring the sequestration of iron, and also they require the growth for virulence. Now, another important element in host defense mechanism of lactoferrin is by binding to certain cell membranes receptors and compete with the infecting viruses from them. So it somehow, these lactoferrin will try to go whenever there is the bacteria sometimes or the virus produce similar or specific receptors in the cell, like the ACE2 that the coronavirus hit. Sometimes the lactoferrin take that shape of that receptor, so it will compete with the virus not to attack those cells. Lactoferrin has actually very numerous areas of action. It's protecting the neonates via breast milk. Also, lactoferrin has been found in the cervicovaginal mucosa in females, reproductive tract, antibacterial, antifungal, antiparasitic, antiviral. Also, it's been found in the airways, it's been found in the mucosa, found in the saliva of all kind, so in the mouth. So it has somehow receptors that bind the lactoferrin, include the CD14, LDL receptors, toll-like receptors, the cytokine receptors, heparin sulfate protease, protoglycan receptors. All these receptors that can be used sometimes by the viruses. Now, the lactoferrin has this kind of ability to attach itself to these receptors, protecting our system from invading viruses. So the entry of bacterial products, or viruses into host cells may also occur via some of these receptors. Lactoferrin acts as an important element in host defense mechanism by binding to those receptors. Well, how the lactoferrin fight the infection? Well, lactoferrin is produced by neutrophils. That's number one. As simple as that. And neutrophils is our actually first sensor of infection. So it produces lactoferrin in order to start its action. Number one, it scavenges iron, thereby inhibiting the bacterial proliferation. Well, as you've seen, each molecule of lactoferrin takes 2 ferric molecules of iron. So this first thing is grab and engulf all the iron in the circulation, so that the bacteria will not find any iron to process or to invade us. Number two, lactoferrin, through binding to its cognate receptors, promotes the maturation, migration, and cell proliferation of many immune cells, including the macrophages, monocytes, which impact on the efficacy of host responses in the course of infection, as has been really documented in many references. One thing very important is regarding the correlation between the lack of immunity and infection. Of course, worldwide, over 40% of children who have iron deficiency anemia are frequently associated with the infection. Anemia, lack of iron. Again, experimental evidence in the last decades show that iron is a fundamental element for normal development of the immune system. Humoral immunity appears to be less affected actually by iron deficiency than in cellular immunity. Then iron is required for monocytes, macrophages, differentiation. Iron is also required for the function of the differentiation of adaptive immune cells such as the T lymphocyte, which is very important. In order for the T lymphocytes to transform into T4, T8, and other producing cytokines, it needs the iron to do that. So if it lacks the iron, that means this function will not take place. So here is the study, a beautiful study that I came across from Egypt. Dr. Tamer Hassan, he's a pediatrician from Medical College in Azi. If you can see the picture and the logo, you recognize where the study was done actually. What he did, he took all children with iron deficiency anemia, and what he just measured, plotted their serum iron and this line and their interleukin 6 level. And you can see here there's a very beautiful strong correlation between the two. The lower the serum iron, the lower is the IL-6. So this figure shows that there is a significantly positive correlation between serum iron and interleukin 6. Now, the significant positive correlation that was observed in their study, as I mentioned, between iron and IL-6 can be explained on the basis of the established reciprocal, which is cause-effect relationship between iron and cytokine production. So there is iron deficiency due to decrease in cytokine secretion, and cytokines actually maintain the intracellular balance. So these two likely work hand in hand in order to fight infection. And of course, if one of them is dropping, the other will drop, and that's why it's very important to correct the iron in these cases when these children with anemia to reduce the infection. So somehow the T lymphocyte, which is a cytokines in iron deficiency, the study reported decreased level of interleukin 6 in patients with iron deficiency anemia due to T cell dysfunction. All right, this is just to correlate these facts together. Now, Jason 2001, removal of iron stores from the body causes a decrease in T cell proliferation and differentiation with subsequent decrease in cytokine secretion. Another study, altered cell marker expression may contribute to reduced T cell proliferation during iron deficiency. Brook 2000 said that iron is involved in RNA reductase and DNA synthesis. So the proliferative phase of lymphocytes and activation is first requiring a step, and it can be diminished during the iron deficiency anemia. Then it said that, and Savondeva said that altered levels of some of interleukins and cytokines actually might lead to immune system impairments in iron deficient patients. So it seems to be that the iron, the intracellular iron effect has an adverse effect of iron deficiency on the red blood cells picture, and growth is well known and was described in several papers. Now, reported in 1985 that depletion of neutrophils count in iron deficiency anemia was found, low leukocyte count resulting from low neutrophils and eosinophils. Iron deficiency is also associated with the impaired ability of polymononuclear granulocytes to kill the ingested bacteria. And neutrophils have many iron-containing compounds, and of course, myeloperoxidase, which is an iron-containing enzyme that is found in the granules of the neutrophils, contribute to the antimicrobial activity, as mentioned before. Now, the cytochrome is another iron-containing compound that is found also specific granules and is required for the oxidative burst that follows the phagocytosis of the bacteria by the phagocytes. So this is where any reaction that hit the bacteria, you'll find iron is there. So it's very important to keep that in mind. Now, the iron is a fundamental element for normal development of the immune system. Now, here we can see that the defects in iron-dependent enzymes, any enzyme that depends on the iron, any defects cause decrease in this oxidative burst activity, which kills the organism inside the monocytes and the neutrophils. Also, decrease the phagocytic activity of the monocytes and neutrophils. It has been found that neutrophils' phagocytic activity was 58% in the anemic group compared with the 74% in the control group. So there is somehow anemic patients have lower actually phagocytic activities. That means their immunity is lower to fight infection. So low immunity due to iron deficiency causes infection. There is a decreased phagocytic and bactericidal activity of neutrophils in iron deficiency anemia. There is a strong evidence for a deficit in the ability of neutrophils to kill the bacteria. A marked decrease was observed also in the killing of certain bacteria. This has been documented in the literature, Staph aureus and Staph albus and also E. coli. This has been associated that this bacteria per se, we're not talking generalized here, these reports correlated actually the marked decrease in the killing of this bacteria, in particular whenever there is actually anemia. Now, reversal of the abnormality was reported within 15 days of iron therapy. This depends on the paper that you read. Some people say 15 days, some people say 45 days, but anyway, iron deficiency patients had significantly lower immunoglobulins, IgG, humoral, and lower actually phagocytic activity of the oxidative burst, which is non-specific, and also lower interleukin-6 of the neutrophils. No significant difference between the patients and controls with regard to the other immunological compounds or ratios. So it's very important that we have seen all this phagocytic activity, actually, and immunoglobulins levels, and also the burst function in the neutrophils, all depending on iron and all being found to be affected in iron deficiency anemic patients. So somehow infection, immunity, and the fight for iron is going hand in hand. And I'll just go through this few slides here to remind you about the ferroportin function and how it really needs to is really working for the purpose of releasing the iron outside the cells and or keeping the iron inside. So it's a very important compound to be expressed whenever it is needed to open the gate or close the gate. Hepsidine and ceruloplasmin are the two enzymes that actually cause the oxidation of the ferrous into ferric in order to open the gate and get the ferric outside to be taken by the circulation. Hepsidine is a very important enzyme that really working on this ferroportin. Either it will actually diminish it or internalize it, phosphorylize it, or degenerate it, and so it will be becoming less expressed, and that means that the gate is closed, no iron will be released outside, or hepsidine sometimes open this gate and make this more expression of the ferroportin, so more iron will be leading outside. This is a very controlling mechanism. Somehow, hepsidine is not only work by demand. When it is needed, there is organ inflammation, but also we found that estrogen has been linked to the work of the hepsidine, and we can see that here in these experiments that ovarectomy in rodents associated with decreased serum iron, and also estrogen treatment reduces hepsidine synthesis in hepatocyte-derived cells. So somehow, whenever there is lack of estrogen, there is also lack of hepcidin stimulation or expression. So estrogen treatment in vitro was also associated with enhanced expression of the HIF alpha and regulates the hepcidin synthesis. So hence its role in the intracellular ion modulation. So somehow, yes, estrogen may work on the same line. Cytokines cause iron deficiency anemia of the hepsidine. So it has been found that in this experimentation, the results of iron accumulation within macrophages and decreased circulating iron levels, which appears to be an effective defense strategy against extracellular macrophages or microorganisms, need to be assessed for iron to be extracted by the pathogens. So it's very important to correlate the two together. Cases of iron availability or deficiency anemia correlating with the liver, actually even acceptance in liver transplants, the phenomena of spontaneous liver allograft tolerance, in which an essential step in the induction of tolerance is the activation and subsequent deletion of liver infiltrating lymphocytes, which really relies on iron completely. Lastly, there is an iron deficiency and response to vaccination. I mean, there has been some reports to say that patients taking the vaccine, but they don't develop the antibodies. Of course, in order to develop antibodies, you need your system to working. But if for that system to work, it needs iron. If you are iron deficient, then more likely your system will not work, and as a result, will not develop antibodies for your vaccine. And this has been seen in many of the measles, actually, vaccines area, and has been reported in the literature over the time. Finally, my conclusion to say is that your immune system is comprised of two parts, specific and non-specific immune systems. Simply, non-specific immunity is your body's first line of defense and refers to a general mechanism your body deploys every day to keep you safe and protect you from disease-causing microorganisms. Non-specific immunity also includes specialized cells that work to find, neutralize, destroy, and or remove the pathogen from your body. Because iron deficiency degrades non-specific immunity, you are more vulnerable to infection and disease and other health complications if you are iron deficient. In fact, frequent infections is a well-known symptom of iron deficiency. And these are my references, and I'd like to thank you very much.
Ladies and gentlemen, I'd like to welcome you back. I hope you enjoyed the first presentation, and I think we need to move on for our next speaker. And I have the honor, honestly, to present to you Dr. Luai Zaid. He's a dear friend and real master in the field of perinatology. He's a consultant of obstetric gynecology in Dubai and United Arab Emirates, associate professor of obstetric gynecology in Egypt, and he had his diploma in fetal medicine from France. I heard him presenting very interesting topics in the past, and I'm really looking forward to his presentation tonight on the role of the lactoferrin in iron deficiency anemia and immunity. Dr. Lai, floor is yours.
Thank you very much, Dr. Shan. I will start by sharing my screen first. So just to confirm, is the screen shared now?
Yes, Dr. I just want to remind also the audience to write their questions in the Q&A box. So
I better be starting. Thank you very much, Dr. Shan, for the intriguing lecture. Honestly, it was a very sophisticated and state-of-the-art lecture about the role of iron in the immune system. Well, I have a thought in my mind. We all go travel long-haul distances just to attend a conference. And in every conference we attend, we're used to hearing lectures like the one just given by, delivered by Professor Hisham, one which is like quite state-of-the-art and cutting edge and like state-of-the-art, needs to be said. But still, we need some basic practical lectures. And so both of them are really quite important and complementary components of what I call conference meals. So you need to know the advances. You must have an idea about the advances in the field, because at least at the end of the day, we are physicians. Some of us are researchers. So we need to know the advances and the molecular mechanisms. That's very important. And from the other side, you need something more basic, practical, and practice changing. So first, I would like to start by thanking Dr. Hisham, thanking the organizing committee and Saja for giving me this opportunity to be among you tonight to discuss lactoferrin, which I chose the name of lactoferrin, the wizard molecule, because it's not only iron deficiency anemia, it's iron deficiency anemia and beyond. So the problem, we have a problem. Yes, we all have a problem. What is this problem? It's anemia. Why is it a problem? We'll see in the next slides. But every single person of us, and I'm here stressing and referring to obstetricians, I think every single obstetrician had this nightmarish case in the delivery room, whether a C-section or a vaginal birth or whatever. This patient was anemic throughout pregnancy, and she bled in the OT. And when she bled, he really wished he could correct this anemia. We all had these patients. We all came across these patients. Some of them, unfortunately, lost their lives for it. Others had severe morbidities and ICU admissions, and that's horrible, least to be said. So we have a problem, and this problem is anemia. Anemia affects roughly 1/3 of this world's population. So every 3 persons living on this planet, 1 of them is anemic. Half of the cases are actually due to iron deficiency. And according to data from esteemed international organizations, iron deficiency is the most widespread nutritional deficiency worldwide. Women seem to be at increased risk for iron deficiency. And I hear a question, why? It's common sense, because they lose almost 60 milligrams of iron on a monthly basis. They have increased iron requirements during pregnancy, and they experience significant blood loss during childbirth. So this was actually an interesting study conducted in Tabuk, Saudi Arabia. The reason it's interesting is that it has been conducted on apparently healthy young Saudi females. And it showed a prevalence of iron deficiency anemia of 12.5%. Because some people may say, well, here in the Gulf area, people eat well, they're in good shape, we don't have this problem. No, we do have this problem. 12.5%, that's a lot. So iron deficiency anemia in pregnancy and childbirth. Anemia in pregnancy is defined, as you all know, as first trimester hemoglobin less than 11 gram%, second and third trimester less than 10.5 gram%, and postpartum hemoglobin less than 10 gram%. Anemia is common in pregnancy. The prevalence ranges from a mere 5% in developed countries to more than 80%, a staggering 80% in developing countries. This number is horrible, 80%, that's horrible. Iron requirements during pregnancy increase dramatically, and this common sense. As the mother's blood volume is expanding, the fetus grows, develops, thus pregnancy should be considered as a condition of impending or existing iron deficiency. So every pregnant woman should be considered in a state of impending or existing iron deficiency. So iron deficiency and the pregnancy outcomes. Well, maternal iron deficiency, it has a myriad of effects. This is just an oversimplification of nature, of what's actually happening. But on the mother, we have increased risk of preeclampsia, wound sepsis, increased mortality, of course. And on the fetus, the low birth weight, the prematurity, intrauterine growth restriction, reduced iron stores. In the offspring, metabolic syndrome and schizophrenia. Yes, schizophrenia. And this is actually something that was quite staggering for me. And I came across this study because I was not actually convinced with the idea of schizophrenic fetuses because of iron deficiency. And this study was actually published in the American Journal in 2020. And they realized that the rapidly developing fetal brain is at particular risk for iron deficiency, which can occur because of maternal iron deficiency. And low maternal gestational iron intake was found in this study to be associated with autism, schizophrenia, and abnormal brain structure in the offspring. Newborns with iron deficiency have compromised recognition memory, slower speed of processing, and poor bonding that persists despite postnatal iron depletion. And this is very serious. We're not talking about a mother who has anemia and you'll give, and that's it. No, we are talking about long-lasting effects on the offspring. The wiring of the brain itself of the neonate may be altered forever, for good. This is important. So this is the problem. Now, the key on how to solve this problem. The key is understanding the iron metabolism. Professor Hisham actually explained it in a fascinating way. I'll just have to stop on like the pivotal points. And these pivotal points are actually your take-home message today. So a revolution actually occurred during the last decade in the comprehension of the physiology as well as in the physiopathology of iron metabolism. Iron homeostasis, you have to understand that the iron system, or the iron homeostasis is iron as a molecule is a very well recycled molecule in the body. There is merely no waste, just a mere loss of 1 to 2 milligrams daily, as you can see in the graph in here. And that's why we need to absorb 1 to 2 milligram daily. As you can see, around 70% of the total body's iron exists in RBCs in the form of hemoglobin and in muscles in the form of myoglobin. And this staggering 70%, as you can see, almost 20 milligram of iron is actually extracted from it through the process of RBC destruction through using the reticular endothelial system, and a similar 20 milligrams plus or minus are again consumed from the plasma pool into the bone marrow, again in the process of RBC production. And we have more or less fixed body stores, myoglobin and respiratory enzymes. All of the system are interchangeable. And approximately 25% is stored as ferritin throughout the body. And finally, the remaining 5% is bound to transferrin, the main carrier of absorbed iron, to different cells. So it's a pretty much recyclable, highly recyclable system in the body. So you only lose almost under normal conditions, almost 1 to 2 milligrams of iron every day. That's why you only need to absorb 1 to 2 milligrams, unless you have iron deficiency. So again, those are the key determinants of iron regulation, and we'll come across those in the next few slides. Here you have the mucosal ends, where food is. Here you have the end products, that's the myoglobin, hemoglobin. Here you have key determinants like the DMT1 and the ferroportin. We'll get back to that. Then the transferrin and the ferritin, the storage and the circulation of the iron, and finally the end products. So this is again the same slide as Professor Hisham actually had, but it's a little bit zoomed. It's the same thing, but it's very important. It's very important. That's the single most important slide throughout the presentation, in my opinion. So let's start with my presentation. So when you ingest iron, iron in your food is actually heme, that's the animal iron, or the non-heme, that's the non-animal iron, and the salt iron, or like the vegetables and plants and cereals. The heme iron is the VIP iron. It has a receptor of its own. It has an entry site of its own. So it has a VIP entrance. That's the heme transport, where it gains direct access inside the cell. And that's why we say heme iron is actually, it has a higher value of the non-heme iron because it has its VIP iron. It has an entrance, VIP entrance. On the other side, if you ingest iron, you usually ingest it in the ferric state. That's the 3+. It has to be reduced into the ferrous state. That's why some of the iron salts in the market, or most of them, are actually in the ferrous form to facilitate the process of absorption of iron. If it's ingested in the natural form in the ferric state, it will have to undergo reduction through the duodenal cytochrome B. This is actually the enterocyte, and this is the brush border, this is the lumen, and this is the basal membrane, and this is the bloodstream. So the iron food is either VIP iron or heme iron, has a VIP entrance to gain access inside the cell. The non-heme iron is usually in the ferric state, undergoes reduction into ferrous state, because iron can only gain access to the cell in the ferrous state, but can only be transported in the ferric state. And that's a stable constant. That's an important concept. It enters through the DMT1 receptors, okay, to gain access inside the cell. So iron is now, whether heme or non-heme, it's inside the cell. Inside the cell, some of the iron will be stored in the ferritin cage, like in storage form, and some of it will exit the cell. The storage form, or the ferritin, the intracellular ferritin, will be lost by shedding of epithelial cells. And this is one of the very important, or the single most important mechanism of iron excretion, is actually by losing cells. That's why iron overload is a problem, because your body has no mechanism of like excreting the iron through the kidneys or through bile. No, the main mechanism of excreting the iron is actually through shedding of epithelial cells of the enterocytes themselves. The rest of the iron it will undergo through the basal membrane. There is the ferroportin, and this is a very important relatively recently molecule that has been or channel that has been recently described, and it is the only, to the best of my knowledge, the only receptor found in human cells that allows iron to exit or allows the efflux of iron through outside the cells. So all of other heme receptors, lactoferrin receptors, DMT1, all of them, they lead to entry of the iron inside the cell. This is the only one that allows the exit of the iron to the outside of the cells. So this ferroportin allows the exit of the iron in the ferrous form, because as we just mentioned, iron can gain access only in the ferrous form, but can be transported only in the ferric form, to be oxidized through the hepcidine into the ferric form, to be transported through the plasma transferrin. This arm is very important. That's where the innovation actually happened in the understanding of the iron metabolism. So this ferroportin is under a strict effect from hepcidine, which is secreted from the liver. Let's talk about this hepcidine, because they refer to it as the master of ironomics. And why is that? Because it's a simple peptide of 25 amino acids, which is predominantly secreted by hepatocytes. And it makes sense because most of the iron you ingested, and when you ingest it through the portal circulation, it has to stop by the liver. So the liver has sensors to detect the amount of iron and to regulate the amount of absorption accordingly. But the hepcidine is not only regulated based on your need and erythropoietic iron requirements. No, because if you are anemic, what will happen is your liver will not secrete the hepcidine, thus releasing the ferroportin in the hepatocytes, in the spleen, and the duodenal enterocytes, allowing the efflux of iron from the cells into the circulation so that it can finally go to its bone marrow, to its end target, that's the bone marrow, to synthesize the RBCs. So anemia is actually suppressor of hepcidine. On the other hand, in case of inflammation or increased extracellular iron concentrations, those lead to increased levels of hepcidine. And when the hepcidine spikes, it blocks the ferroportin, thus trapping the iron inside the cells, preventing its efflux into the circulation. It's something similar to the insulin and the glucose, something like that. So how much iron do I need? Well, the only important thing that you need to remember is men only need 8 milligrams of iron a day, because you usually absorb 10% of the iron you're ingesting, more or less. And if you ingest like 8 to 10 milligrams, you'll end up absorbing the 1 to 2 milligrams that you need, that you lose every day. Women, on the other hand, nearly need double of that, and pregnant women need triple that. That's what we need to remember. Pregnant women need triple what an adult male needs from daily iron. Again, as we just said, the heme iron, that's the animal food. The non-heme iron is the plant food, and plus eggs, of course. This slide is very important. It's actually very important because it gives you the spectrum of changes that will happen from normal to iron deficiency anemia. What we need to understand here is that iron deficiency anemia is the end of a spectrum, so that your body starts suffering from the very beginning, and it goes through a series of compensatory mechanisms. When they all fail, you have overt iron deficiency anemia. This concept is very important because if you understand this concept, we know that to correct iron deficiency anemia, you have to go backwards. So you have to restore all of these steps, but backwards. So you have to treat the anemia, and when you treat the anemia, you have to replenish the stores. So as you can see here, in a normal person, you have normal ferritin, you have normal transferrin or total iron binding capacity, and you have normal erythropoietic parameters, the MCV, the MCH, and of course the hemoglobin. When you start having the anemia, the iron deficiency, the first marker that gets affected is the serum ferritin, and that's very important. All other markers are normal. It's just your stores are getting depleted. The more you proceed, the more the ferritin, the more of other markers and compensatory mechanisms start appearing in your lab result. So at the beginning, you have the ferritin and the stainable tissue iron. Most probably you won't detect that. Then in the stage of iron deficiency, you have decreased ferritin, you have no stores. The transferrin saturation itself decreases, which means the reciprocal of that is the total iron binding capacity, which will increase, which means the total iron binding capacity, your ability of your body to bind iron. When it increases, it means that there is not enough iron in your transportation system. So to understand this simply, you have iron in the target product that's in the end product, that's the hemoglobin. Then you have iron in the root, the transportation, and you have iron in your stores. The first thing that gets depleted is the iron in your stores. Then the iron in the transportation, that's the ferritin. Then you have problem in your iron, depletion of the iron on the way, in the roots, that are transferrin saturation and the total iron binding capacity. Eventually, at the end, when all of these fail, your hematopoietic parameters become affected. And the first one is the mean corpuscular volume, the MCV, which begins to dip. Because your bone marrow, what it's trying to do is, it needs a certain budget of iron to synthesize high quality hemoglobin. If it does not have this budget and you give it less iron than expected, what your bone marrow will do to compensate is to make an intermediate quality RBC. That's by decreasing the MCV. The more depletion that happens, the worse will become the RBC until the full-blown picture of microcytosis, hypochromia, and low hemoglobin. So the first step you should do after, in case of anemia, is to do the ferritin. If you have low ferritin and you have low hemoglobin, it means that you have iron deficiency anemia. If you have normal parameters but low ferritin, it means that you are in the stage of iron deficiency and not iron deficiency anemia. On the other hand, if you have high ferritin with low hemoglobin, it may mean that you have anemia of inflammation. And if you have high ferritin and low MCV, you have to remember the thalassemia, whether major, minor, or trait. Also, a very important message is you always have to remember that serum ferritin is also an acute phase reactant that spikes in cases of severe inflammation. And the best example now is the COVID patients. When they do the ferritin, it's almost always shooting. So now we have to just, in a couple of slides, just to stress on the fact of anemia of inflammation. Well, it's the synonym of anemia of inflammation, or AI, is the anemia of chronic disease, regarded as the most frequent anemia in hospitalizing chronically ill patients. Most probably, we don't encounter these patients in the obstetric population, but still they do exist. They are not that frequent, but we do meet them more or less on a daily basis. Patients with lupus, patients with chronic renal disease, all of these are, and they may suffer from anemia of chronic disease. It is prevalent in patients with disease that cause prolonged immune activation, including infection, autoimmune disease, chronic renal disease, cancer, and obesity. And that's why it's important, because it's on the rise and you have to keep it in the back of our minds. Inflammation-induced cytokines stimulate the release of the master regulator of iron, the hepcidine. So this inflammation will need to increase in the levels of hepcidine. And what hepcidine does is it blocks the intestinal iron absorption and cause iron retention in the reticular endothelial cells, resulting in iron-restricted erythropoiesis. If you remember the slide where there's the hepcidine and the ferroportin, the hepcidine, when it rises because of inflammation, when its levels increase because of the inflammation, it will block the action of ferroportin, leading to trapping of the iron intracellularly, depleting the iron transportation system from its iron cargo, thereby finally depleting the end organs, which are the bone marrow, from the iron that it needs to perform its erythropoiesis function. The diagnosis of anemia of infection is a diagnosis of exclusion, but unlike an iron deficiency anemia, there's a tip or a hint in detecting it, supported by the presence of hypoferriemia, so I have decreased iron, but increased ferritin. So this should trigger your attention that this is a case of anemia of inflammation or anemia of chronic disease. So now we know the problem, now we know the key on how we can solve this problem. What's the plan? Pregnancy recommendations as per the Royal College is to do full blood count. It should be assessed at booking and at 28 weeks. That's important. All women should be given dietary information to maximize iron intake and absorption. And for anemic patients or women, a trial of oral iron should be considered as the first-line diagnostic test. So if you come across a pregnant woman at 14 weeks, 15 weeks, booking lab, she has mild anemia, she has moderate anemia, it will not harm to do a trial of oral iron for 1 month, then to reassess the situation. So those are the recommendations. Also from management recommendations include, as prevention strategy, diet and pregnancy precautions. Also, the iron supplementation as per the British Society of Gastroenterology guidelines. It suggests that all iron deficiency anemia patients require iron supplementation, that parenteral iron can be used if oral preparations are not well tolerated. The third step and the management of these conditions are blood, and blood transfusion should be reserved for patients who are at risk for or who have cardiovascular instability due to their anemia. And this would never happen in a healthy female in the reproductive age group unless she has chronic illness and underlying morbidity, or if her anemia is quite severe, that's below 7 grams per cent. Erythropoiesis stimulating agents like the erythropoietin are not recommended for patients with mild to moderate anemia and either congestive heart failure or coronary heart disease. So the first step, that's prevention. I'm sure you're all familiar with this, so I don't have to waste some time, waste more time on it. In the IV iron therapy, some pregnancy-related conditions requiring IV iron therapy, like inadequate response to oral iron therapy. You all have seen patients like this. You give her like oral iron preparation after the other, and at the end of the day, it's like she's like now 33, 34 weeks, and hemoglobin is like 9.5. You don't have time. You don't want to put patient into risk. And at this point, you may need to give the IV iron. Patient non-compliance, intolerability to oral iron, and other factors as well. So the last step, which is the iron supplementation, and here where we raise the first question, which is what is lactoferrin? What has got to do with iron supplementation? I'm sure you all know by now, thanks to Professor Hisham's presentation, but it won't harm to go through it quickly again. Lactoferrin, which is known as lactotransferrin, is a multifunctional glycoprotein of the transferrin family. Yes, it is one of the transferrin family, which is responsible for transferring the iron. It's found in varied secretions of organisms, especially cow and human milk. Colostrum, we all know colostrum, the first milk produced after baby is born. It contains high levels of lactoferrin, almost 7 times if compared to mature milk. Lactoferrin iron deficiency anemia. So that's the immature form, or the apolactoferrin, and this is the iron saturated form with 1 molecule chelates 2 ionic iron molecules. Why lactoferrin and not traditional iron salts? If you remember when we talked about the enterocytes and the lumen and the hepsidine, the ferroportin, let's start to see what's more that lactoferrin can add. Well, lactoferrin improves utilization of endogenous iron and improves iron absorption. Some even postulated that there are lactoferrin receptors. Remember the VIP receptor, or the VIP entrance for the heme iron? Some researchers actually suggested that there is a similar VIP entrance to lactoferrin bound to iron, and that maximizes the utilization of iron. It has, again, the second thing is it has natural anti-inflammatory substance, which is able to modulate hepcidin and ferroportin synthesis through the downregulation of interleukin-6. So if you can see here, if you remember, the hepcidin, which blocks the ferroportin, interleukin-6 actually is a marker of inflammation. It's an important marker of inflammation. Interleukin-6 signals that there is an inflammatory, reaction going on in your body. And this will trigger the liver to increase the levels of hepcidin, to block the ferroportin, trapping iron inside the cells, to deprive the pathogen or the aggressive host, the aggressive pathogen, from utilizing this iron to multiply and to combat your body. And so this interleukin-6 is a marker of inflammation, which will increase hepcidin, leading eventually to decrease iron efflux from the cells into the circulation. Lactoferrin has an anti-inflammatory property that will actually block this hepcidin action. It has minimal GIT side effects, the lactoferrin, of course. It's highly resistant to GIT proteolytic degradation due to its partial iron saturation, and it has high but reversible iron affinity. A myriad of studies have been performed comparing lactoferrin versus the ferrous sulfate, which is the traditional salt in most of the commercial available forms, salts, like this one. This study is actually published in 2016 in the Journal of Maternal Fetal Neonatal Medicine. It showed that lactoferrin was more effective than ferrous sulfate, that's the salt, over a 2-month period in pregnant women with iron deficiency anemia, with fewer GIT effects, side effects, and better treatment acceptability by patients. Again, this one, the ferrous sulfate, this study is quite interesting because it showed that ferrous sulfate, that's the traditional salt in iron supplements, increases interleukin-6 and fails to increase hematological parameters and pro-hepcidin. On the other hand, the bovine lactoferrin was shown to be more effective and safer alternative than ferrous sulfate for treating iron deficiency anemia in pregnant women. This was actually a meta-analysis which showed that in iron deficiency anemia with pregnancy, bovine lactoferrin was better tolerated with higher increases in hematological parameters when compared to conventional oral iron therapy. This was a famous meta-analysis published in 2017. So, and this one last thing, this last study conducted on the efficacy of lactoferrin oral administration in the treatment of anemia of inflammation. And that's a very strong point in favor of the lactoferrin, because as we saw in the previous study, just before the meta-analysis, the ferrous sulfate was shown to increase the interleukin-6, which is a marker of inflammation, which will stimulate the release of hepcidin, leading to blocking more ferroportin and trapping of iron intracellularly, preventing its absorption and depleting the bone marrow from it. So in a way, according to that study, ferrous sulfate may have some kind of paradoxical action. It is iron, so it helps that the cells get the iron inside them. I'm talking about the enterocytes, but in the same time, its side effect is that it increases the interleukin-6, which is a marker of inflammation that traps the cell inside the cells. Yes, the iron is absorbed, but it is trapped inside the cells. So it does not go, the end result does not go to the bloodstream, and so it's useless. Lactoferrin, on the other hand, shows a greater benefit and efficacy than standard ferrous sulfate management and can be considered as a promising compound in treating anemia, and especially anemia of inflammation, through its ability to downregulate interleukin-6. This has been proven in meta-analyses and in several studies. So it downregulates interleukin-6, thus releasing the negative feedback inhibition exerted by hepcidin on ferroportin, thus restoring ferroportin-mediated iron export from the cells to blood in a hepcidin-dependent or independent pathway. So when you take the lactoferrin, it has an anti-inflammatory action, and this action specifically releases the ferroportin from the suppressive effect of the hepcidin. And that's a strong point for lactoferrin in cases of anemia of inflammation or anemia of chronic disease. So let's move on to the next slide. And the following section, I call it the wizard molecule. So we have discussed the lactoferrin with iron deficiency anemia. So are there any other benefits of lactoferrin? Well, all of these are actually functions, biological functions of lactoferrin. To date, about 20 different physiological roles of lactoferrin have been reported, including iron binding, transferring, antibacterial, antiviral, antifungal, anti-inflammatory, and anti-carcinogenic properties. Lactoferrin also seems to be involved with regulation of bone marrow function. That's the myelopoiesis we all know about. And it seems to be able to boost the body's defense immune system, as Professor Shan actually mentioned in his presentation. So let's take some of the famous or important or relevant functions of lactoferrin. Actually, Professor Shan referred to something very interesting. That's the iron wars. So that's the lactoferrin on the left, which is competing with the siderophores from the bacteria here on the right on the iron molecule. And usually the lactoferrin helps, wins in most of the instances in preventing the iron uptake by the bacterial proteins at the siderophores on the right. So lactoferrin's primary role, is to sequester free iron, and in doing so, remove essential substrates required for bacterial growth. And it also binds to lipopolysaccharides of bacterial wall through the receptors, because bacteria, in their attempt to scavenge any iron in any form bound to anything, they have lactoferrin receptors. And the lactoferrin acts as like a booby trap or a dynamite. So it binds to the lipopolysaccharide of the bacterial wall, and the oxidized iron part of the lactoferrin, because this lactoferrin has iron, And the bacteria, in its way, in its trial to have this iron, to acquire this iron, it has lactoferrin receptors. So the lactoferrin bound to iron binds to the lactoferrin receptor on the surface of the lipopolysaccharide bacterial wall, and the oxidized iron actually oxidizes the bacteria by formation of peroxides. And this affects the membrane permeability and results in the cell breakdown. So that's the antibacterial activity, or at least part of the mechanisms understood that would describe the antibacterial activity of the lactoferrin. The antiviral activity, that here it becomes more interesting, because the most studied mechanism of antiviral activity of lactoferrin is its diversion of viral particles from target cells through binding to the same cell membrane lipoprotein used by some viruses to penetrate a cell. So some viruses, they use some receptors to bind to the cell membrane. Lactoferrin competes with these viruses by blocking these receptors. Lactoferrin also was shown to hydrolyze RNA by exhibiting the properties of ribonucleases. Now it gets interesting. Sorry. The SARS-CoV. The SARS-CoV, in attacking cells or in infecting cells, the mechanism, one of the very well-described mechanism was that the virus itself, it rolls, as you can see in the top in the image A, it rolls onto the cell membrane by binding to heparan sulfate proteoglycans, which are these tree-like stuff, these stemming branching stuff. So the virus, it kind of rolls to speed down and to slow down, using the heparan sulfate proteoglycans as speed breakers, so that it can find eventually its target, which is the angiotensin converting enzyme 2 receptor. So what happens is the virus rolls onto the cell membrane by binding the heparan sulfate to the speed breakers and starts scanning for specific entry receptors, which leads to subsequent cell entry. Lactoferrin was shown to block the infection of SARS-CoV by binding the same speed breakers, the heparan sulfate proteoglycans, preventing the preliminary interaction between the virus and the host cell and the subsequent internalization process. So the question is this also apply to SARS-CoV-2? And the answer is, it seems yes, because the supplementation with lactoferrin, other supplements, could have a role both in preventing SARS-CoV-2 infection and in mitigating the clinical course in infected patients, contributing to the prevention of immune-mediated organ damage. And in vitro data support lactoferrin as an immune modulator of the antiviral immune response with moderate effects against SARS-CoV-2 infection. So those were actually published in 2021. So those are quite recent publications describing the potential role of lactoferrin in minimizing or decreasing the deleterious effect of SARS-CoV-2, or at least decreasing the infection itself. Again, the lactoferrin and an antifungal activity. Yes, the antifungal properties were related to its ability to damage fungal cell membranes and alter their permeability, and also to iron chelation. So similar to the antibacterial mechanism. The native human lactoferrin showed a higher activity against Candida as measured by the MIC, that's the minimum inhibitory concentration, than the form with high iron saturation. For the neonatal immunity, as we mentioned before, lactoferrin is the second predominant protein in human milk, the colostrum, if you remember, with higher concentrations in the colostrum, 5 to 6.7 milligram per ml, if compared to mature milk, 0.2 to 2.6 milligram per ml only. Lactoferrin has several physiological functions to protect the gastrointestinal tract of the neonate, and actually a large systematic review has shown lactoferrin to be a promising agent for reduction of late onset sepsis and necrotizing enterocolitis. One of the important roles and promising areas in the domain of the multifactorial biological functions of lactoferrin is its potential anti-carcinogenic effect. The anti-cancer activity of bovine lactoferrin has been demonstrated in experimental lung, bladder, tongue, colon, and liver carcinogenesis on rats, possibly by suppression of phase 1 enzymes such as the cytochrome P450 1A2. Even in dermatology, I was actually personally overwhelmed by seeing this, but it was actually a relatively recent 2017 systematic review concluding that there is encouraging evidence to suggest that lactoferrin may be beneficial in acne, psoriasis, and diabetic ulcerations. So to conclude and wrap things up, Anemia affects roughly 1/3 of the world's population, with a significant impact on maternal and neonatal morbidity. Please remember the brain wiring of the kids, that's important. Half of the cases of anemia are due to iron deficiency. Lactoferrin may be considered as a strong addition to our armamentarium of therapies against both iron deficiency anemia and anemia of inflammation, and that's a stronghold. That's a strong point in favor of lactoferrin. To date, about 20 different physiological roles of lactoferrin have been reported, including iron binding, transfer, antibacterial, antiviral, antifungal, anti-inflammatory, and anti-carcinogenic properties. I thank you for your attention. I hope it was not a lengthy lecture, and thank you very much. Thank you.
Right. Thank you very much, Dr. Lai, for this excellent, beautiful presentation. I'd like to go through the questions and answers now from our audience here, and see a question here, I think, coming my way. Many COVID pneumonia patients are developing an unexplained severe anemia. Is it AI, anemia of inflammation? If yes, what is the treatment? Yes, it is AI, as we all know. There is a burst of cytokines and all these things coming out, and they block that hepsidine. So for that purpose, you know, I mean, we know the reason behind anemia here. Usually they say treat the cause, okay? The underlying cause. Definitely you have to treat that COVID or pneumonia, whatever, in all its aspects, as you all know, with different medications available, antiviral, etc. However, if you think like scientifically, if you know that the hepcidin is the cause here, so why don't we have something that inhibits the hepcidin? Well, yes, it's been thought of, but unfortunately it's not available yet. I mean, it's been manufactured, it's being tried in a few trials, but we're still waiting for that. So in order to wait until we have something really definite that will work and safe to inhibit hepcidin, I think the best thing is to go by, for example, the status of the anemia. Maybe you use blood transfusion. You can use, for example, the intravenous iron. And also we have lactoglobin, which is really can be supplemental in this situation. Do you agree, Dr. Lai?
I totally agree. Totally agree, Dr. Shan. Totally agree.
Okay, so the next question is going is actually for you here, and it says, is lactoferrin helping in patients with gastric sleeves or bypass?
Well, these patients with sleeve gastrectomy or bypass, they definitely have some degree of impairment in iron absorption. And so for these patients, if I personally would prefer putting them on lactoferrin, especially in cases of pregnancy, as a mechanism of helping the body to overcome the defective mechanism of absorption. Because as Professor Shan highlighted earlier, stomach is a fully functioning stomach is very important in iron absorption. So if you take part of that outside the equation, most probably the patient may develop anemia long term. So you have to help the body. So you took something from the mechanism of iron absorption. So you have to help the body with a different mechanism. I think lactoferrin fits well in this indication, personally.
Okay, I can try on this question. We know that iron absorption increase when you have anemia. Does iron absorption rate increases during pregnancy? Well, actually, it does not. You know, I mean, with anemia, it's not the absorption rate, because we have seen that patients who are pregnant and their hemoglobin actually is dropping, and they cannot take the iron because maybe there is with all these GI symptoms related to the iron intake, so they remain anemic. So there is no compensation here, even if you increase the dose of iron. And nowadays, we don't really advise this. We have to stick to the 60 milligram elemental iron in one single dose daily, still you are not going to increase the absorption of the iron in the gut. So all what you need actually is just to monitor this patient, check her diet. There's one of the questions talking about what is the daily requirement. Dr. Lai mentioned to us that in normal women, you need about 18 milligrams daily, while for a pregnant woman, you need at least 27 milligrams per day of iron taking through the diet to replenish their daily requirements. So I think that's the thing. It's a matter of monitoring it, and then you go for intravenous iron, or you go for lactoglobin in case of. And here, actually, I find lactoglobin as an alternative for those who are having GI symptoms related to the oral iron intake. So if she's one of those kind, then you tell her, okay, take away those iron pills and take the lactoglobin, which really it does not really sensitize your stomach or your GI. Dr. Rai, there's a question here. If hemoglobin is normal, but ferritin is low, do these patients need intravenous medication or oral medication is enough?
Well, I think they don't. Actually, IV iron is something very invasive. For example, I think in the first trimester, it is not indicated, it is not recommended to give IV iron. So IV iron is something very invasive. Try to let the body heal itself as long as you have time. If you don't have time, that's a different story. But in this patient, the hemoglobin is normal. So which means that the patient has only iron deficiency and not iron deficiency anemia. So you clearly have time. So give the patient oral medication. You still have time. There is no problem. The end product is being manufactured in a good way. She has good hemoglobin. She only has low iron stores. So if you leave her like this, she will progress through the end of the spectrum, eventually developing anemia. So on this page, the answer is in these medications, I think oral medication is quite enough.
The same person came back with another scenario here. Do you advise high-risk people in dangers of COVID and even other infections to get standard supplemental lactoferrin and vitamin C? Yes, both of them work fine in situations of infection, inflammation, and I think there is no harm of taking those medications or drugs. A question here, other questions regarding the lactoferrin, similar like other oral iron preparations, and take iron with vitamin C, avoid the alkali food. Supplement or food contains Ca, okay, or take. Can you read that? I don't.
The questions regarding lactoferrin, similar like other oral iron preparation, intake iron with vitamin C, avoid to alkali food supplement or food contains. I'm not sure I understand the question. I'm sorry, but I think, yeah. So I think she's referring to the interaction between the traditional iron salts and the food and the alkali and the acidification, because the ferrous sulfate you have to take, or the iron salts, you have to take them with like vitamin C or acidifying agents to promote the absorption. And I think that's what she means. But I mean, lactoferrin is, I think it's independent from this pathway, and it does not interact with these pathways. It's a glycoprotein. It traps iron, but it itself, it's a glycoprotein, because there's actually one of the questions referring to, should we give lactoglobin to patients who have lactoferrin, I mean, to patients who have lactose intolerance? And the question is, of course, yes, because it's a glycoprotein. It is not lactose. It is a glycoprotein, unless the patient has specific kind of allergic reaction or hypersensitivity to the lactoglobin itself, which is quite extremely rare. But yes, you can give it to patients having even lactose intolerance.
We always tell patients not to take calcium and iron tablets together. Does this advice applies here with lactoglobin and calcium tablets? Well,
to be honest, I don't like pragmatic questions. I mean, what would be harmful to the patient to tell her, please take the calcium alone? It may chelate part of the iron before the lactoferrin gets the chance to trap it. So why do that? Just let's do the right thing. It will not hurt her. to give to put the calcium in a meal and the lactoglobin in the separate meal, lactoferrin in the separate meal.
Exactly. Is antacids effect on iron absorption, affecting the iron absorption, antacids, Dr. Ulai?
Of course, yes. They are chelating it. They may chelate part of the iron. So it's the same thing. Tannic acid, that's the T. Milk products. I mean, like you have to, when you take, if you're taking iron salts, they have high affinity to interact with their environment. That's why you give acidifying agents like the vitamin C, and we refrain from giving chelating agents such as calcium, such as antacids. But when you are talking about different forms of iron which are not known to interact with their environment or poorly interact with their environment, like the heme iron, yeah, that's not an issue. But as a general rule, iron salts, they do interact. As for the lactoglobin, it's a glycoprotein. I wouldn't like to put it into test by giving a chelating agent that competes with the lactoglobin in trapping the iron. Why do that?
Here there are two questions about intravenous iron. Carboxymaltose can be given in pregnancy. Yes, it can, but it should be given in the second or the third trimester, not the first. And talking about giving it fast, yes, you should give it fast over 15 to 20 minutes. You should not really giving slowly. A question for you, Dr. Lai, does vitamin D levels affect the bioavailability of iron?
I think indirectly. And please correct me if I'm wrong, Professor Hisham. I think indirectly, because low vitamin D levels, they may impair the immune function, and the impairment of the immune function may invite some kind of infection. So it's like in an indirect way, but as a direct mechanism between iron absorption and vitamin D absorption, to the best of my knowledge, I don't think so. I don't know, Professor Hisham, if you have something to add. What's your opinion?
Well, I'm very pragmatic about vitamin D, honestly, levels, because I tell them we always find low levels of vitamin D's, and they start to correlate it with infertility, pregnancy, and all these things, anemia. No, it's not a cause, and it shouldn't be really correlating low levels of vitamin D with whatever thing in medicine. I think it's still an area that is hazy and depends on the laboratory methods that you're using, and it's also the patient themselves. So if a patient has both iron deficiency and thalassemia minor, can we give iron injection or oral? So I'm talking about iron deficiency. Can we give the iron injection?
For me, I would never give iron injection in a thalassemia patient. I would never get, because thalassemia is actually chronic hemolytic anemia, and they have functional iron deficiency, and we can look at thalassemia as anemia of chronic disease in a way, but there is some mechanism of iron overload in these cases. I would rather give lactoferrin to these patients, definitely not IV iron, and orally I prefer to give them first lactoferrin and not oral iron, at least at the beginning, unless I have really strong, clear evidence with the help of a hematologist that this patient needs iron supplementation.
Actually, they're talking about the lactoglobin here alone, and when to consider iron supplementation along with it. Would you combine ferrous sulfate plus lactoglobin at the same time?
Personally, yes, sometimes, yes. If the patient is anemic, because lactoglobin, what it does is it helps recycling the iron, it helps utilization of iron itself. And so, yes, these combinations, sometimes I do these combinations. So I can give an iron salt alongside the lactoglobin.
Right. Somebody asking about the use, why we're giving intravenous iron fast. Well, actually, the reason here is to to abolish or abort any sensitization process. I mean, it's those who are giving these on a longer period of time. These are the people who are more likely to get anaphylactic shocks and being sensitized by the drug. So. I think we have maybe one more question to take before we close. This is for you, Dr. Lai. Is specifically third trimester woman may have low iron, and how to correct sickle cell anemia? Two questions here.
Just two different questions. Well,
Yes.
yeah, third trimester women may have low iron, of course, because the third trimester women, they have the maximal fetal consumption. The fetal needs are increased. The fetal placental unit itself, it's increased. Fetal blood volume is around 80 to 100, 110 ml per kg. That's the fetal blood volume. So the more, the bigger the fetus, the more the fetus to grow, and the more kilos he weighs, the baby weighs, the more will be his blood volume. And this blood is hemoglobin, and it simply needs iron. So definitely women in the third trimester should be considered in a way iron deficient. That's true. And they are most at need for iron. And that's actually one of the reasons the Royal College recommends of testing the iron around 28 weeks. So at booking and at 28 weeks again.
Right.
That's part of the question. And how to correct sickle cell anemia? Well, that's high-risk pregnancy, and I think it's beyond the scope of this lecture, but because sickle cell anemia, you have different types of crises, the chest crisis, sequestration crisis, the oxidative stress. I mean, like, it's a topic on its own, sickle cell anemia. I don't think it's, I think it's beyond the scope of this lecture.
Can IV iron be started earlier in pregnancy? And when we know a pregnant lady cannot use the oral replacement, even when there is no anemia. As I mentioned before, in only the second trimester, you can start the intravenous iron. There are some reports about trying the first trimester, but still need to be further documented. Fecal lactoferrin and calprotectin as marker inflammatory bowel disease or infection. Any comment, Dr.
Well,
Lai?
calprotectin is actually one of the markers of the inflammatory bowel disease. And fetal lactoferrin, again, is a sensitive and specific marker in identifying inflammation in chronic IBD. It's non-invasive. It's very useful as a screening tool. But the calprotectin with the initial, I think the cutoff is 200, I think. And that's like one of the gold standards in the diagnosis. But fetal lactoferrin, again, it's a sensitive screening tool.
If a patient is having anemia for a long time and living with it, like no symptoms, should we correct that, especially if she is getting pregnant? If so, is there any special regimens? Well, I think yes. If she's considering pregnancy, we know there are side effects and untoward adverse outcome of anemia in pregnancy, affecting the prematurity, affecting also the APH and perinatal outcome. So yes, of course, we should correct the anemia prior to pregnancy. Especially if she needs, for example, intravenous, you can start it before she gets pregnant. Otherwise, you cannot give the intravenous the first trimester if she's severely anemic. Then we have a question. Usually we depend CRP procalcitonin for detect infection. So what about hepsidine? Will it be a screening test? Will you use hepsidine as screening test in anemia?
Again, that's very pragmatic. I mean, if I don't have any other tests, yeah, I would look for hepsidine, but we're not talking here about Down syndrome or something that needs a lot of tests and double marker and stuff. You just do CBC. You have low hemoglobin, you do the ferritin. You have low ferritin, and then you have iron deficiency anemia. There is no point of testing for the hepsidine. I mean, it will not add value. Yeah, there has been some researches about hepcidine guided screen and treat interventions against iron deficiency anemia and stuff like that. But I mean, like, it's still in the research arena, not actually in the everyday practice, because it's highly variable from lab to lab. It needs like really stringent criteria, and I don't think it's useful. I don't know what do you think, Professor Shan, but I don't think it's useful.
No, I don't think it's useful. I don't think it's the time actually to look into this, because we know once we diagnose anemia and we know the reason behind anemia, that's enough. You just correct that iron requirements. Hepsidine itself is not really a marker because it's just a partial mechanism that may actually affect the outcome or the status at that moment. Can we use lactoglobin alone in iron deficiency anemia, and anemia of inflammation above, with the hemoglobin above 9?
Listen, I think there's some kind of confusion in this area, but so let's put it this way. I think that lactoglobin as a natural glycoprotein, which is present in all the body secretions, it will not harm to give it maybe as a prophylaxis against iron deficiency. If the patient, despite taking the lactoglobin, or a patient who's not taking the lactoferrin and she starts developing anemia, at this point, we may need to add iron supplement alongside the lactoferrin. So if I have a patient with a hemoglobin of 9.5, I will not give him lactoferrin alone. Lactoferrin, it maximizes the utilization of the iron, but it's only 30% saturated with iron. So you need to give the patient iron itself so that lactoferrin can help this iron through the VIP passage and the VIP entrance inside the cells. That's what I think.
Nicely said, Dr. Lai. Actually, I have enjoyed your presentation, your contribution to this question, and I really like to thank all the audience. I mean, we have more than 500, mashallah, attending this webinar session. I'd like to thank Zaid and Anahita for their technical support, taking us from the beginning to establish a webinar of this kind. Thank you very much, and I hope we sometime we meet you soon in one of those webinars in the future, inshallah. The CME will be available in 7 days. So please fill out the evaluation form and answer all the webinar quizzes that will be sent to your registered email ID to obtain the CME credits. All right. So the webinar will be available on demand on the YouTube MCI Middle East YouTube channel. And the address is below there. And okay, here is there's some information or announcement coming up shortly. Thank you very much, everybody. Hope to see you again. Thank you, Dr. Lai.
Thank you, Professor Shan. Thank you very much, sir. Thank you. Thank you, everyone. Have a wonderful evening.